So we're going to take a look back at this myofibril. This is a zoomed in picture of a myofibril. Notice we've got this light band right here. This light band is also called the I band, and it has a dark midline, and that's going to be because of this Z disc here. The Z disc, of course, is where the actin filaments are attaching themselves to this midline in which they can attach. But because there's a lack of myosin filaments in this area, it's going to give it the it's gonna make it look like it's a lighter area. This is called the A band, and we're going to take a look at it in a moment, but that is going to be a darker area because of the myosin filaments. If you'll notice, so this Z disc here, if you go from Z disc over to Z disc, that's going to make up what we call a sarcomere. And just one sarcomere is a unit. It's like a contractile unit on this myofibril. And of course, you've got sarcomere after sarcomere after sarcomere just lined up and stacked up against each other. As I was mentioning earlier, this is the A band here in the middle. This is what makes up the dark part of the striations on striated muscles. There's also this H zone here in the middle, and the H zone is going to be a lighter area because take a look at this. I've got my actin filaments right here. My actin filaments don't go all the way into the H zone. So in the H zone, you've got myosin filaments in here, but you're lacking actin filaments. So right here, it's going to be really dark because you've got both actin and myosin, but right here in the middle, you're lacking the actin filaments, so it's actually going to be slightly lighter. It says, notice that the I and A bands are responsible for the striations of skeletal muscle tissue. When we took a look at this stuff in the tissues unit, and we took a look at skeletal muscle, and we saw that those are striated. This is the reason that those muscles are striated. You've got a light band here due to the presence of only actin filament. You've got dark bands here due to the presence of both actin and myosin. And actually what's going to happen when this muscle contracts is that these actin filaments are actually going to dip into this H zone, and then it's going to go back. But Miss Johnson's going to talk about that with y'all in the physiology section. But it's just really important to know right now that the striations are formed because of the actin and the myosin filaments. So I want to take a look, a zoomed in look at just one sarcomere. Notice here's our Z disc right here. Here's our actin filaments right in this area, and then these dark red pieces are our myosin filaments, and these are actually packed in together, and this is going to make up what we would call a contractile unit, and this is going to be the thing that actually causes the contractions in a muscle to happen. Thick filaments are going to be these red filaments here, these myosin filaments. They're going to contain myosin protein. That's why they're called myosin filaments. They're also going to contain ATPase, which is an enzyme which is going to help split ATP. And of course, we have to have lots and lots of ATP in order to generate power for these muscle contractions. These myosin filaments are going to form our A band, which is our dark band. And they've got these little bitty projections on them, which we call myosin heads. And so what's going to happen is these myosin heads are going to reach out, they're going to grab hold of these actin filaments, and it's going to form what's called a cross bridge when a link is formed here. So the thick filament is actually going to reach out and grab the thin filament, and then it's going to pull it in and then release it back. And that's how the actual contraction happens. Like I said earlier, Miss Johnson's going to go over that with y'all when y'all get to the physiology portion of the notes. Here's those myosin heads I was talking about. Each one of these, of course, is attached to a myosin protein, and the myosin heads then can reach out and grab onto an actin filament, and they pull back and forth and fun stuff like that. Thin filaments are going to contain actin protein. They're also going to contain tropomyosin protein filaments and a troponin protein. The tropomyosin protein filaments are actually going to act as a receptor site blocker so that when these myosin heads are not engaging, those receptor sites are going to be blocked. Once a myosin head reaches out to grab onto one of these actin filaments, then that tropomyosin protein is removed and a connection can be made there. Of course, these thin filaments are anchored to the Z disc, which is the end of the sarcomere. Remember, this is what separates sarcomeres. And it says the zone where filaments do not exist in the sarcomere is called the H zone. So this is the H zone here, and this is where you have only myosin filaments, but you have a lack of actin filaments. As I was mentioning earlier with the actin filaments, this is the tropomyosin protein here, this orange piece. And these little blue things that look like olives, this is the actual actin protein. So what's going to happen? You can see these little holes here, which would be like the pimento part of the olive. What's going to happen is the tropomyosin is going to be removed. A myosin head can reach in, grab hold here, and then it's going to pull back and forth to cause a contraction to take place. As long as this tropomyosin is in place, though, no connection can be made. So the tropomyosin has to be removed before a myosin head can reach in here and grab hold to form a cross bridge. And that's going to be the end of the anatomy of the muscle portion. Like I said, Ms. Johnson is going to continue on in the next video. Thanks.